chapter. So take it away, Miranda. You're muted.
Cool. Can you hear me? Good?
You're good.
Yeah, so let me minimize that. Today I'll be talking about, as Juliana mentioned, one of my dissertation chapters that's currently in review, titled Rhododendron Seedlings Achieve Similar Performance Across Light Environments with Anthocyanin Accumulation and Architectural Adjustment Under High Light Intensity. So my entire dissertation chapter focuses on the species Rhododendron minus, which is an evergreen shrub native to the Southeast United States. And it's really interesting to study from a light perspective because, as you can see here on this image in the left, we have Rhododendron minus growing on these rock outcrops in the Appalachian Mountains, which presents a very stressful environment as well as highly exposed, meaning that the plants are likely to be subjected to high light intensity. And if light absorption exceeds what can be safely processed in the photosynthetic pathway, then that can damage the plant and cause serious issues, potentially even cell death. So from this image, you can see these individuals likely are exposed to high light intensity, but you could also imagine a scenario where seeds are dispersing down the mountain into different areas that may be more shaded. And so this species, it was expected that this species could grow and survive under a range of light intensities, likely adjusting physiology and morphology to accomplish that. So looking at this image, again, of an adult Rhododendron minus there growing again in the Appalachian Mountains, we can see some hints that there may be a need for photoprotection going on. So again, helping protect the plant from damage acquired through excess light energy. So one, you can see these red colored leaves. It's from a pigment known as anthocyanin, which offers photoprotection through two main ways. Number one, it offers some antioxidant properties, and two, it can help take in some of that excess light so that the photosynthetic pathway can maintain that efficiency. And two, we also observe this steep leaf angle going on, which is opposite of what we saw from the first talk, where in Thermonasty, leaves exhibit this pendant leaf angle. In this image, under high light intensity, we were expecting potentially to see a steep leaf angle to, again, help minimize light from being absorbed, rather than if the leaves were more perpendicular to that incoming light source, more light would be absorbed. So these leaves having a steeper leaf angle are more parallel to those direct sun rays. So with this in mind, for this work, we asked, do rhododendron minus seedlings exposed to higher light intensity have more anthocyanin, with the idea that this pigment is going to offer some photoprotection? Less chlorophyll, with the idea that chlorophyll being the main pigment responsible for light absorption, if there's less chlorophyll, then there would be less photons then being sent through the photosynthetic pathway, and hopefully then the plant would be able to keep up with the amount of light that's being taken in. And then lastly, leaf movements to reduce light capture. Where here, originally I was thinking, maybe steep leaf angle, like in that photo from the previous slide, or potentially a greater degree of leaf overlap, which could induce greater self shading to again reduce that light capture. So this experiment was focused on the seedling stage. Again, going back to the idea, if we have seeds being dispersed into more shaded regions, it likely is very important that, especially at the seedling stage, the plant can handle a variety of different light intensities. So back in 2012, Steve Krebs collected seeds from this location called Hawksbill Mountain in North Carolina. Then all the way fast forward all the way to 2023, where we germinated some seedlings for the experiment. I then created a well draining soil mixture to eventually pot the plant into trays to then be subjected to our light treatments. So this experiment was conducted at Holden Arboretum, and we had 3 light treatments. In the back here, you can see we had trays that were placed under shade cloth as our shade treatment. We had seedlings placed under supplemental light, which was our light stress treatment. And then we had our control treatment, which was just seedlings exposed to ambient light or natural greenhouse conditions. So after the experimental setup, I had to figure out, okay, how am I going to go about estimating or measuring anthocyanin and chlorophyll, which were our 2 pigments of interest for this work. And rather than doing traditional chemical methods with pigment extraction, I wanted a more high throughput method. So I landed on building what's called this multispectral imaging device. Based on this paper here, published in Sensors back in 2024. It's a fairly complicated device, but the main idea is that you can get a non-destructive estimate of both chlorophyll and anthocyanin based on the amount of light reflectance under different color LEDs. So light reflectance off the plant helps us to then calculate in formulas, indices for both anthocyanin and chlorophyll. And this is a great method to help compare pigment concentrations across plants under different light treatments. So for our first question, do seedlings under higher light intensity have more anthocyanin? From our multispectral imaging device, we estimated anthocyanin through NDAI, or normalized difference anthocyanin index, where a higher value means relatively more anthocyanin. And we did see significantly higher NDAI under supplemental light, which also matched observations of increasing redness with increasing light intensity. So 2 lines of evidence there that suggest that these seedlings are producing anthocyanin under light stress. So then we asked, do seedlings under higher light intensity have less chlorophyll? And for chlorophyll, we used what's known as NDVI, or the normalized difference vegetation index, where a higher NDVI means relatively more chlorophyll, and a lower NDVI is relatively lower chlorophyll. So we were expecting to see the least amount of chlorophyll in our supplemental light treatment, but actually we saw no significant effect on NDVI. So this suggests that plants across these light treatments examined here can maintain similar levels of chlorophyll. Now, what's interesting about this is that potentially what's going on is that these plants in the supplemental light treatment were actually maintaining their chlorophyll levels because of the photoprotective benefits of anthocyanin, almost acting as a shield for the chlorophyll to help minimize that excess light energy absorption. And similar to how we observe similar chlorophyll levels across plants, we also saw similar levels of total biomass, which makes sense if plants are maintaining the similar amounts of chlorophyll. Likely they were photosynthesizing at similar rates and producing similar amounts of biomass, so that at the end of the experiment, they all were similar in size. So in addition to pigments, we also looked at leaf movements over time, and this was led by an intern that I was working with named Emma Farley. And for this, we calculated change in projected surface area as projected surface area final, which was measured at the end of the experiment, minus projected surface area initial, which was measured towards the beginning of the experiment. And this just shows one leaf at one time point, but just to give you an idea of how we got these projected surface area values, Emma photographed the plants from an aerial perspective and used ImageJ software to help create a mask of the plant and get out that area again at 2 time points, the difference being our change in projected surface area. So we asked, do seedlings under higher light intensity have leaf movements to reduce capture? To orient you to the graph, we have our change in projected area on our Y, and a negative value would mean a decrease over time, which is what we would expect under our higher light intensity, and an increase in projected area over time is represented here by this positive value. And that is indeed what we see, where we see our shade and ambient mean is positive, whereas under supplemental light, we see a decrease or a negative change in projected area. So looking over here at this example of a seedling under supplemental light, looking at our initial and final, we can see at the end of the experiment, there was actually new leaf growth. So that should theoretically increase our projected surface area. But with this degree of leaf drooping and greater leaf overlap, we actually see a net decrease, which should help minimize light interception. So going back to our research question, do rhododendron minus seedlings exposed to higher light intensity have more anthocyanin? That is what we found, but we actually found that they have similar levels of chlorophyll across treatments, and we did see leaf movements to reduce light capture. Now this work, again, was only conducted on seedlings where the seeds were collected from one location in the Appalachian Mountains, but as you can see here of this, which is represented by that blue circle. But as you can see here, with these distribution points of rhododendron minus, this species spans a range of latitudes and elevations, which creates climate gradients. So we were wondering whether there are differences in pigments across the range due to climate differences as well as solar radiation differences with latitude. So then we asked, do plants from locations with higher solar radiation and or more stressful climates, i.e. colder and drier, exhibit higher leaf anthocyanin content, with the idea that anthocyanin is a well-rounded pigment that not only helps with light stress, but also can help act as an antioxidant for damage occurring under stress related to climate. So for our first question, do plants from colder locations exhibit higher anthocyanin content? Here you can see a map of 6 plants from 6 locations that had been growing at Holden Arboretum. Plants from our northern colder locations are shown in blue, and our southern warmer locations shown in these reddish pink. To orient you to this graph, we have mean annual temperature, which is the temperature extracted at each of these locations based on weather station data. And then we have our NDAI, again, where a higher NDAI means more anthocyanin. So again, we're expecting plants from colder locations to exhibit higher anthocyanin content, and that is indeed the relationship that we find. Next, moving on to plants from locations with higher solar radiation also exhibit higher anthocyanin content, again with the idea that anthocyanin will help with photoprotection. We actually see that over here on this end of the graph, our locations with higher solar radiation actually had lower NDAI. So this seems to contradict what we found in our greenhouse experiment on the seedlings from one location, where we see increasing anthocyanin with increasing light intensity. So what potentially could be going on here? Why the difference between our greenhouse experiment and this experiment on plants originating from different locations across the range? Well, first of all, this measure of solar radiation does not account for fine scale heterogeneity in the light environment. So, for example, plants from colder locations generally tend to grow on rock outcrops, which again are exposed, stressful, generally high light environments, contrasted with plants growing at warmer locations in the southern end of the range. Those tend to grow in the understory where it may be more shaded. So the fact that solar radiation isn't accounting for these habitat differences may be at least partially what's going on. But likely more importantly is that in addition to being in these very exposed locations, the winters are very cold. And so photosynthesis is going to be more downregulated in our cold locations than our warm locations. And hence, light absorption may more easily exceed photosynthetic capacity when it's cold. And so the cold locations generally were found to have more anthocyanin, likely because of a greater need for photoprotection. I have a lot of people to thank, but I definitely want to shout out my advisor, Juliana, not only for supporting me in every step of the way, but also for putting together this lovely convention. And I'll be happy to answer any questions.
Thanks, Miranda. We have time for a couple of questions. We're running a little behind, but we have a break coming up, so we'll go ahead and take some questions if anybody's got one before we move to the next talk.
Miranda, good talk. Just a quick question. Have you come across any studies where they've looked at the actual leaf anatomy as also one of the factors when it comes to dealing with high light, which exceeds the photosynthetic efficiency of the plant? In other words, especially palisade layers. There's a one layer versus multiple palisade has been often invoked in such scenario.
Yeah, I think from what I'm remembering is that, the greater amount of palisade layers that could confer some degree of photoprotection with, you know, um, sunlight being absorbed in certain layers versus other, and it's almost a filtering effect. So
Mm-hmm.
that actually gets at the idea of leaf thickness, where if you have greater leaf thickness,
Mm-hmm.
that could confer some photoprotection. So we actually looked at specific leaf area, which gets at that variable. I didn't have time to talk about those results,
Mm-hmm.
but we actually found that differences in specific leaf area um, may have been more likely due to an unfortunate drought effect of that soil that I created since it was so well draining. So I don't believe that we saw an actual true light treatment effect on SLA or any sort of leaf thickness, but potentially either with greater degree of light intensity or maybe just a different species. But that's a great question.
Mm-hmm. I mean, you have those populations from the higher elevations. I mean, the northern and more colder and warmer climates. It might be a good idea to compare the anatomy of those. In our work, we found that one of the species of Rhododendron ponticum actually, which does not have thermonasty like Catabians does, but has some of these other features to handle the light. We believe one is palisade layers and also a double layer of epidermis as opposed to single
Mhm.
layer in Catabians. So some of those things will all contribute that, like you say, to the thickness of the leaf. Yes.
Yeah,
Yeah.
that's
Good.
great.
Good.
Great. Let's do one more verbal question and then you've got another question to answer. So Peter Norris asked, did you account for variable sun, as in angle, question mark?
So at least in terms of our greenhouse experiment, it was conducted in a greenhouse. So theoretically, greenhouses are designed to scatter the light. So hopefully there was even distribution of sunlight, but we also replicated our treatments across 3 greenhouse benches with the idea that, maybe the angle of sun was weird for one treatment. That's why you replicate it 3 times to spread out that error, if that answers it at all.
Yeah, and I think putting my advisor hat on, someday we'd like to look at more of the variability in light within the natural environment, but we're not field biologists, so somebody collaborate with us. Awesome. Miranda, do you want to answer? There's one more question in the chat, if you can type the answer, and I think what we'll do is we'll move on to our next speaker.
Great, thank you.
Our next speaker is Dylan Nakaji Conley, who is a research specialist at the Holden Arboretum. And we're going to continue with the R- fund. So take it away, Dylan, and share